The NADH and the FADH2 molecule that produced from the previous stages of aerobic respiration contains high amounts of energy. So, in the oxidative phosphorylation, the main purpose is to use the energy that released from the electron flows along the electron transport chain to produce ATP. Oxidative phosphorylation involves electron transport chain and chemiosmosis. Let's begin with the electron transport chain, which means a series of electron carrier molecules that will be found in the cristae or crista of mitochondria, which is the folded inner membrane of mitochondria, and the electron carrier molecules will carry the electrons from NADH and FADH2 through reduction and oxidation reaction, and finally produce ATP via chemiosmosis. Of electron transport chain will be found in the inner membrane of mitochondrium, which is a structure that encloses the matrix of mitochondrium, which is also the fluid field space of this organelle. The first component is complex 1, which is also called as NADH dehydrogenase. It's a transmembrane protein. The second complex, which is complex 2, is called as succinate dehydrogenase, is partially embedded in the inner membrane of mitochondrium. The next is coenzyme Q, or ubiquinone, the only lipid-soluble electron carrier of ETC. It can be found fully in the hydrophobic tail regions of the inner membrane. The next is complex 3, which is cytochrome C reductase, and followed by the cytochrome C, which will attach at the hydrophilic head region of the inner membrane. And lastly is the complex 4, which is cytochrome C oxidase. The name of each complex 1, 2, 3, 4 implies their function in the ETC. Oxidative phosphorylation produces ATP by using the energy released from energy flow along the electron transport chain. And this is because each electron carrier, they have different free energy, which is summarized in this graph. For example, the NADH, which is the electron carrier produced from glycolysis, link reaction, and Krebs cycle, they carried high amounts of free energy, which made them unstable, and they have the lowest affinity for electrons. Means that the attraction, okay, for electrons is the lowest. So they have the higher tendency to donate the electrons to the first complex in the ETC, which is complex 1. When complex 1 accepts the electrons from the NADH, it is reduced. And at the same time, NADH is oxidized because they lose the electrons to the complex 1. So energy is released when the electron is passed from the high energy NADH to the down here complex 1. And now when the complex 1 accepts the electrons, it has slightly increasing in their free energy. So they become unstable. Okay, immediately they will pass the electron to the next electron carrier, which is the ubiquinone. So now complex 1 is oxidized because they pass the electrons to ubiquinone, and ubiquinone is reduced because they accept the electrons from the up here complex 1. Whenever the electron is passed from high energy molecule to a lower free energy molecule, the energy is released during the electron flows. And redox reaction occurs because one compound is reduced and another one is oxidized simultaneously. And this redox reaction and the electron flow will repeat until the electron is accepted by the oxygen molecule, which has the lowest free energy, or they have the highest affinity for electrons. So oxygen will combine with the two protons from matrix of mitochondria and produce water molecule. Study the pathway of electron transport from NADH, which is the electron or hydrogen carrier that produced from glycolysis, link reaction, and Krebs cycle. The NADH is the high energy molecule that will donate the electrons to complex 1, which is the NADH dehydrogenase. Redox reaction occurs because NADH is oxidized to become NAD+, and complex 1 is reduced when they accept the electrons from NADH. And the electron carry energy will make the complex 1 increase in their free energy. But when they show increment of the free energy, the complex 1 is unstable, so they will pass the 2 electrons to the next electron carrier, which is the coenzyme Q or ubiquinone. So redox reaction occurs again because ubiquinone becomes reduced, and the energy is released during the electron flow, and this energy will be used to activate the complex 1 to pump proton from the lower concentration, which is the matrix of mitochondria, to the higher hydrogen concentration in the intermembrane space. Coenzyme Q will pass the high energy electrons to the next electron carrier, which is complex 3. And now the coenzyme Q becomes oxidized and complex 3 becomes reduced, which means they become unstable because they accept the high energy electrons. So they will pass the electrons to the next electron carrier, which is cytochrome C. The flow of electrons will release energy, which will be used to activate the next proton pump, which is the complex 3, to pump the proton from the lower concentration, which is matrix of mitochondria, to the higher hydrogen ion concentration, which is the intermembrane space. And now the reduced cytochrome C becomes increasing in their free energy, which made them unstable, so they will pass the electrons to the last electron carrier of ETC, which is complex 4. So redox reaction occurs again because cytochrome C becomes oxidized and complex 4 becomes reduced. Complex 4 will become reactive and pass the electrons to the oxygen atom, and each will combine with the 2 protons from the matrix and becomes one water molecule. The passing of electrons will release energy, and this energy will be used to activate the last proton pump in the ETC, which is complex 4, to pump proton from matrix of mitochondria to the intermembrane space. Another source of electron is from FADH2, which is the molecule produced during Krebs cycle. FADH2 contains lower free energy compared to NADH, and this made them pass the 2 electrons to the complex 2 instead of complex 1. Redox reaction occurs when FADH2 is oxidized to produce FAD, and complex 2 is reduced when they accept the electrons from FADH2. When complex 2 becomes reduced, they have the high energy electrons make them unstable. So they will pass the electrons to the next electron carrier, which is ubiquinone or coenzyme Q. And redox reaction occurs again. The reduced coenzyme Q is unstable, and then they will pass the high energy electron to the next electron carrier, which is complex 3. When complex 3 is reduced, they have the high energy electrons. They will immediately pass the electrons to the cytochrome C. So when the electrons flow from high energy electron carrier to lower energy electron carrier, the energy is released. Same goes to the electron flow from complex 3 to cytochrome C. It will release energy, and the energy will be used to activate the proton pump, which is also the complex 3. So proton or hydrogen ion will be pumped from lower concentration, which is matrix of mitochondria, to higher concentration of hydrogen ion, which is the intermembrane space. Now, the reduced cytochrome C carrying the high energy electrons will become unstable. So they will pass the electron to the last electron carrier in the ETC, which is complex 4. So redox reaction occurs again. And complex 4 will pass the electrons to the last electron acceptor, which is the oxygen atom that will accept 2 hydrogen ions that found in matrix of mitochondrium and produce a water molecule. So when the electrons flow happens from high energy molecule or high energy electron carrier complex 4 to the lowest energy electron acceptor, which is the oxygen, extra energy is released during the electron flow, and it will be used to pump the hydrogen ion from matrix of mitochondria to the intermembrane space. Before we study the chemiosmosis, which is the last step of oxidative phosphorylation, let us have some idea about the relation between the ETC and chemiosmosis. So during ETC, both electron carriers or hydrogen carriers, which are NADH and FADH2, will be oxidized by passing their electrons to the components of electron transport chain. So the electrons and the hydrogen, once they have been released from the NADH and FADH2, the electron will be passed along the ETC through redox reaction. The flow of electrons will release energy, and the energy will be used to activate the proton pumps, which are actually complex 1, 3, and 4. And the proton is pumped from the mitochondria matrix to the intermembrane space. So the accumulation of protons in the intermembrane space will create a hydrogen ion or electrochemical gradient, which able to generate proton motif force. When the proton is bumped by the complex 1, 3, and 4 from the matrix of mitochondria to the intermembrane space, they will cause the higher concentration of hydrogen ion in the intermembrane space, and this produces a gradient between the intermembrane space and the mitochondria matrix. So they will produce the proton motive force. The force will drive the movement of hydrogen ion from the intermembrane space to the mitochondria matrix, because this is a passive transport that occurs at the ATP synthase, which is an enzyme found in the inner membrane of mitochondria also. The diffusion of hydrogen ion or proton from intermembrane space to the mitochondria matrix will drive the ATP synthase to produce ATP by combining the ATP with the inorganic phosphate group. And this process is called as chemiosmosis, because the chemicals, which is the hydrogen ion, will move from higher concentration to lower concentration, like the osmosis we have studied in semester 1. So with the theories of electron transport chain and chemiosmosis, we can predict the number of ATP molecules produced from each electron carriers. Let's say we start with NADH. NADH will pass the electrons to the complex 1, which is the NADH dehydrogenase. And the electron will flow along the ETC until it is accepted by oxygen atom that combine with 2 protons to produce water molecule. So if we focus on the pathway of electrons flow from NADH, it's able to bump the hydrogen ion or proton from matrix of mitochondria to the intermembrane space 3 times, because the proton pump involves complex 1, complex 3, and complex 4. Each time of activation of proton pump will generate the proton motive force, which means it's able to drive the hydrogen ion that near to ATP synthase to move down the concentration gradients, which means they will move from the higher concentration, which is the intermembrane space, to the lower concentration of hydrogen ion, which is the matrix of mitochondria, along the ATP synthase. This is an enzyme that will catalyze the production of ATP. So, theoretically, we predict that one NADH will be able to produce 3 ATP through oxidative phosphorylation. Another source of electrons is from FADH2. As we understand, according to their free energy, FADH2 will bypass complex 1, which means they will pass their electrons to complex 2 instead of complex 1 first. So the electron pathway begins from complex 2 to coenzyme Q or ubiquinone, complex 3, cytochrome C, complex 4, and the electron is finally accepted by oxygen atom and reduced with the 2 protons to become water molecule. So the electron pathway from FADH2 will cause 2 times of activation of proton pump. So they will generate the gradients of hydrogen ions 2 times, or they will generate the proton motive force 2 times that enable the movement or diffusion of hydrogen ion along the ATP synthase 2 times. So chemiosmosis will occur 2 times to produce 2 molecules of ATP. And theoretically, we predict that one FADH2 will produce 2 ATP. So now we look at the total of ATP molecule can be produced from one glucose. As stated in the beginning of this topic, ATP can be produced by two modes, which are substrate level phosphorylation and oxidative phosphorylation. Substrate level phosphorylation can occur during glycolysis and Krebs cycle. In the glycolysis stage, from one glucose is able to produce 4 ATP through substrate level phosphorylation. But at the beginning of glycolysis, which is the energy investment phase, 2 ATP are used. That's why there are only 2 net ATP produced during glycolysis. And another 2 ATP are produced during the Krebs cycle, where the succinyl coenzyme A is converted into succinate. Now we look at how the 34 ATP can be produced via oxidative phosphorylation, which is summarized here. Okay, this is because from one glucose, starting from glycolysis until the Krebs cycle, is able to produce 10 NADH, which include 2 NADH from the glycolysis state, 1 NADH during the link reaction, but because we start with 2 pyruvate here, that's why we have 2 NADH. And we have 6 NADH from the Krebs cycle, if we refer to one glucose. So total we have 10 NADH. Each NADH will produce 3 ATP during the oxidative phosphorylation. So we have total of 30 ATP. And we look at another electron carrier, which is FADH2, which are only produced during the Krebs cycle. So from one glucose, it's able to produce 2 pyruvates and 2 acetyl-CoA that complete 2 times of Krebs cycle. So we have 2 FADH2. And the 2 FADH2 will involve in the electron transport chain and chemiosmosis, which is the stages happen in the oxidative phosphorylation to produce 4 ATP. This is why we have 34 of ATP produced via oxidative phosphorylation. So compare these two modes of ATP production. Oxidative phosphorylation play a very important role here, since that they can produce much more ATP compared to substrate level phosphorylation. So from one glucose, the total of ATP can be formed is 38 ATP.